No, kitty cat. Wait, you leave that alone. Okay. All right. Good day, everybody. So, sorry, my trusty companion is insisting on some love. He doesn't like being picked up, though. So, what we're going to be doing today is just going over and recapping the work that we did on RNA processing, the end of last lecture, because there's quite a lot there to take in. And then, so that's really kind of more of a ask questions and or clarifications, and I'll walk you through. I don't really want to teach the same thing all over again, right? Because I've already done that once. And then after that, we're going to get on to translation, and there's a bunch of stuff going on in translation, particularly, well, there's mechanism, right, which is, it's not complex, but there's a lot of stuff going on. There's, I mean, it's kind of complex actually. There's the genetic code, right? The how mRNA is translated into amino acid sequence. And then finally, like some peculiarities essentially of prokaryotic gene expression, right? Which will have implications when we talk about control of gene expression in prokaryotes later, right? So understanding the basic mechanism of translation in prokaryotes will help you understand the mechanism of how they control gene expression, right? So they're very much tied together in prokaryotes, at least. Eukaryotes are again different. Okay, so essentially, do we have any questions on RNA processing? Let me just bring up that PowerPoint from way back here. All right, because this is obviously, it's a big chunk of the material, and we have an exam coming up on Thursday. So even, hang on, that's weird. Why are you not picking up my, okay, advanced then? No. Sorry, I'm just trying to get the PowerPoint up, but it doesn't seem to be. All right, I just have to share my screen then, I guess. Okay. Yeah, it's going to be I mean, mostly this is the exam, right? You know, transcription and translation and control and gene expression, not control of, but just gene expression in general. Let me get the chat back up. So now's a good time to get into that. Good question. No is the answer. So those are really there as examples of lots of different types of RNAs. I don't actually know what, well, I think snRNA stands for small nuclear RNA, and we do cover those. Those are part of the spliceosome. Snow RNAs, I don't even know what they are. Actually, let's look up what are snow RNAs. I learned something. Small nucleolar RNAs. Damn, yeah, I don't know anything about those. miRNA is microRNAs. That is something that we'll be looking at in control of gene expression. So it will be in the kind of what we cover in the course, because we're going to be really looking at RNA stability, stuff like that, but we're not going to be covering that in this particular exam. That will be in exam 4. So yeah, small nuclear RNAs, snRNAs, I'm pretty sure those are small nuclear RNAs. Yeah, there you go. So snRNAs are part of the spliceosome. So if we go back to, where are we? So these are SNRPs. Where we got? Do we have a definition of that somewhere? I thought we did. Oh, there we go. Which are basically a mix of small nuclear RNAs or snRNAs and proteins, which create our small nuclear riboprotein particles. Those are essential. Well, those make up the spliceosome, right? So they all do different jobs. We've got some which recognize the star of the intron, called one of the splice acceptor sites. We have some which recognize the branch point, and that helps hinge the intron over to form that lariat loop. There are some that catalyze the, I'm not sure, I guess it is a phosphodiester bond, which you can see here, right? So essentially, this is like a simplified version, but the hinge point, let's say, is around where the N is in intron. There's going to be, and I don't know the numbers of all these, I'm not going to test you on that, but you do need to know kind of essentially what these things do. Come on. Oh, it's U2, right? So loop that intron back over after it's been cut, right, at the 5' splice site, which is what another SNRP does. And then a different SNRP, again, is going to form this phosphodiester bond here, right, between the 5' phosphate of like the end, the original 5' end of the intron, and another base, or the 2' OH of another base in that. And so that's what causes that lariat loop, right? So that's like a rigid permanent structure until it gets degraded later on. Right, so again, I'm not going to expect you to memorize what U1 does and what U4 does and stuff like that, because at this level, that's just not important. But you do have to understand the process of, you know, recognizing the intron, because obviously you can't splice it out if you don't know where it is, right? That's the GUAG consensus, right? Or GUCAG, right? So those are nucleotides, RNA nucleotides, obviously, bases, which are just inside the intron, right? Like the first two, or I mean the consensus sequence here is 6 bases, but you get the idea. The first few and the last few bases of the intron, right? So that's how the SNRPs know where that intron is as it gets produced by the RNA polymerase. And then essentially it's just the process, right? You got to cut it, you got to bend it, you got to stick it, cut it, and then stick it some more, right? And that's what this spliceosome as a complex does, right? Splice sites recognized, cut, bent over the branch point with the help of a different SNRP. The 5' end is stuck to the branch point, right, to create that stable structure. Exon 1 is kind of kept in the fray, right? It's carried along by another SNRP, and then you finally cut out the intron, and then you stick exon 1, the end of it, to the beginning of exon 2, right? That process you need to have a general conceptual overview of. So yeah, snRNAs, yes, or really in the context of SNRPs and the spliceosome. There are all kinds of colors. Any of the ones that are labeled U and then a number, those are SNRPs. So remember, what's that? Small nuclear nucleoprotein. There you go. That's why we call them SNRPs. It's an absolute pain to type it out for. Yeah, and it's an absolutely critical part of transcription in eukaryotes. It's hard to stress how important this is because, and I think I'm pretty sure I talked about this last lecture, there's two big issues with not removing an intron. So one thing is that it doesn't code for protein. Well, I mean, it has RNA bases, right? So if it were included, it would code for protein, but it would be protein that shouldn't be there, right? Amino acids that shouldn't be present. So at the best, you're going to get a whole bunch of gibberish in the middle of your protein, right? If that intron is not removed. And the second reason is that what's in the intron is not constrained by natural selection, right? So if you have a mutation in the exon, then, and that changes amino acid sequence, then that may be acted against, or maybe for, but most likely against, by natural selection, right? It would be a deleterious mutation. There's no such constraint in the introns because they don't code for proteins. And so if you read into an intron, there's almost certainly going to be a stop codon in frame with the frame and the exon. And we'll talk about frame and stop codons and stuff like that in a little bit when we talk about translation. But essentially, you won't just get, the intended bits of protein with gibberish in the middle, it'll just stop, right? And so typically, if you have mutations, where are we at? Come on, back, right? In this consensus sequence, right? It's called splice acceptor mutations. Then that is almost always like 99.99%, whatever, loss of function. So you will not get any functional protein from that version of the gene, right? So there's quite a lot of reasons why introns got to be removed. It doesn't necessarily have to happen in a linear way though, right? So you could have intron 2 spliced out before intron 1 gets done. It doesn't really matter, but as soon as they appear from the RNA polymerase, they are available to be spliced out. And it just, gets done at, at some point before the mRNA is fully processed. Okay, other questions? Hopefully you will all understand what 7-methylguanine cap is, or 5-MPG cap. If you don't, you should. You also need to understand what its purpose is for a couple of different reasons, actually. One of which we've covered, which is stability. The other one, which we'll cover in a little bit in terms of translation. You also need to know about the 3' UTR, again, for similar reasons, stability and translation. So you really need to get the whole kind of picture of RNA processing in eukaryotes. Okay. Questions? Anybody got any questions? You can chat them out, or you can just shout them out. It doesn't really matter. I'm not too fast. Yes, this test is on Thursday. It is, I'm pretty sure, yeah, so we're going to start gene reg on Thursday. So that will be the lecture that we have on Thursday. And then the exam will be available 12:15 onwards until midnight, as for exam 1 and 2. Wait, leave that alone. But essentially, what is on the exam is up to the end of the lectures today, right? So nothing on Thursday will be in the exam on Thursday, if that helps. So yeah, it's kind of keeping up the pressure. Unfortunately, it just doesn't let up until, you know, July 28th, when the final is. And the final is not cumulative. The final is on gene regulation, biotech, and genomics, right? There's no, well, I mean, they're inherent, as I say this to every group of students, the last exam in a course is inherently a little bit cumulative anyway, because, you know, we build up to those topics that we cover at the end of the course, but it's not explicitly cumulative, right? I'm not going to be asking you specific facts which you found only in the first 3/4 of the course, right? Cool. Questions? If not, I can get started on translation. Please do take the opportunity to ask questions, though. It is the very best way of improving your grade. Ah, very happy kitty cat. When I'm not teaching on Monday, Wednesday, and Friday, she just spends the whole time yowling at me to come up and sit here so that she can get, you know, petted for however many hours I spend sitting here with you lot. She's a very happy cat then. Okay, so if no more questions, and if any do occur to you, that's totally fine. Just, you know, stop me and I'll cover those, and then just resume the other bit. I'm not particularly bothered by that. Okay. Well, anyway, it's good to see that you survived your personal experience in The Shining, Angela. Hopefully you don't have any internal or external parasites that you wouldn't want to talk about. Peter, you know, once, where was it? It was Medford, Oregon, I think it was. We were supposed to go camping, but my wife was scared that we were going to get eaten by a bear. You know, this was near Crater Lake in Oregon, which is a really cool place. Anyway, we stayed at this Holiday Inn, and I don't know what was going on. Yeah, yeah, I would too. It was we had a good long argument for like 2 hours that it took to drive from Crater Lake to Medford. But there was this like rhythmic kind of squeaking banging from the roof, like the room above us, and it certainly wasn't you know, a couple being very friendly with each other, because it went on all night, like without any change in the tempo. I don't know, maybe someone was running a printing press or something up there. But yeah, none of us got any sleep, and they didn't even have breakfast either, which was, you know, add insult to injury. Yeah, that sounds pretty janky. Anyway, a good test of a like a scary, like bad motel is when you get in the shower, you want to make sure you don't touch the sides of the bath or the shower cubicle. That's pretty good, you know, extreme. Okay, so where are we at? We've finished up. So this is just a general overview of process, right? And generation of the mature transcript, right? And it's very important to remember there's a big difference between pre-mRNA and mRNA. Pre-mRNA is mRNA that's still in the process of being finished, right? Process of being processed. And mature mRNA is this, right? 5' cap is there, introns are removed, polytail has been added, or poly-A tail, sorry. As soon as that happens, it can be exported out of the nucleus into the cytoplasm. Okay, so just as a refresher, nice hand-drawn picture here. This is the anatomy of a mature mRNA from a eukaryote, right? Remember, we have our cap, we have our tail, we have our, this is ORF, which stands for open reading frame. I don't think we have that, so let's type that out. Oops. Hang on a sec. And CDS equals coding sequence. So essentially the same thing, just different words for it. And we still have these untranslated regions at the beginning and the end, the 5' UTR and the 3' UTR. These are important in translation. So the 5' UTR is essentially where a lot of the translation machinery assembles. And this is also the, actually, yeah, bacteria mRNAs do have UTRs. Yeah, I'd have to double check on that. And the 3' UTR is kind of, it's involved in the control of translation, right? The modulation of translation, and also the control of mRNA stability, right? So typically, this region of the transcript is where those small microRNAs, right, which is something we'll talk about in a little bit next week, bind and result in the degradation of the mRNA transcript, right? So these two regions, even though they don't code for protein, are very important in the overall production of protein in terms of the control of gene expression, right? So again, you know, we kind of build on stuff. We'll be building on that information when we talk about control of gene expression in eukaryotes. Okay, so there's a lot going on in translation, right? So let's give you a general, I got some new pens. Actually, I need to put these somewhere else because I don't think I put the, let's use this one up anyway. It's my Scottish heritage, you know, never waste anything. You probably tell from the beard, right? Throwing like cassocks and stuff around. So essentially, oops. It's the same kind of deal for, oh yeah, thanks, good point. Actually, someone I saw made a Expo Revitalizer, which was basically an Expo holder on the end of a motor shaft, and it just span it really, really fast, so all the ink went back down to the tip, which I thought was cool. So there's initiation, right, which is basically ribosome assembly, and also finding the start of, God, that my writing looks like shit. Hopefully it'll be more legible when you see a picture of it. Got to find the start codon, and we'll talk about what the start codon is in a second. This is different between eukaryotes and prokaryotes. Prokaryotes go straight for the action, essentially. And that also ties into the differences in the transcript itself, because prokaryotic mRNAs do not have a 5' cap. Eukaryotes, they do like a scanning mechanism, right? So this process very much differs between prokaryotes and eukaryotes. You have elongation, and that is with charged tRNAs, and we'll talk about what charged means in this sense. It's not like you stick them in the battery charger. It means that they have amino acids attached to them. And I'm going to put little sparkles around this. And GTP. So GTP is essentially the sister of ATP. It's often used in cellular mechanisms as a source of energy, right? And actually, it's actually a signal as well. Like can switch between different states, or proteins can switch between different states, depending on whether or not it's hydrolyzed. Not so much the case here, but it probably gets into it somewhere. I'm not going to go there because I don't really fully understand the mechanics myself. And then we have termination, which reminds me, I need to watch all the Terminator films for my kids. I'll be back. And so that's all about stop codon, damn it, and the release factor. Right, so that's basically what we'll be going over in terms of the mechanism, right? There is other stuff, like I mentioned before, that we'll need to cover. In particular, the translation part with the tRNAs, right? There's some important stuff there in terms of degeneracy, right? The genetic code, how that specificity is maintained, right? And it needs to be. And also, there are some pretty fundamental differences between eukaryotes and prokaryotes, aside from, you know, the various nuts and bolts in here, right? And that really ties into how prokaryotes, not always, but many times, controls the expression of the genes that they have, right? So this is our kind of general plan, at least for the mechanism. So I'm going to be bouncing around the PowerPoint a little bit. And the reason for that is largely because it's an inherited PowerPoint. And so in the way I think about things obviously differs from the person I inherited it from. I haven't got around to redoing all of them yet because it's a pretty massive job. So let's talk about initiation. Well, let's actually talk about some of the things that are required, right? So this, where are we at? Oh, it's really weird that it won't let me share individual applications anymore. Essentially, there's a bunch of stuff, right, which we're going to need. We're going to need, obviously, the mRNA processed in eukaryotes or not in prokaryotes, as the case may be. We're going to need, where are we at? Here we go. Ribosomes. And so ribosomes are, they're pretty weird things, right? So both prokaryotes and eukaryotes have ribosomes which consist of two main subunits, right? There are other bits and pieces around, but typically you have a small subunit and you have a big subunit or large subunit. That's what they're called. And they have S numbers, which stands for Stromberg. It's how they sediment in a centrifuge. I don't need to worry about that too much. But basically, the bigger the number, the bigger the protein, right? So you have the small one, which is typically assembles first, and then you have this big one which comes on. And it's the big one that has the catalytic activity. And that catalytic activity is provided by an rRNA. So rRNA equals enzyme. And the fancy name for that is a ribozyme. And it catalyzes, I'll move this over a little, formation, actually, two things. Removal of amino acid from the tRNA and the formation of peptide bond. Right, so in a sense, the protein, right, part, which you'll see here, right, so this is a small subunit, this is the large subunit. They're in many ways a scaffold, right, a way of maintaining everything in the right context so that these reactions can be catalyzed by the RNA, the ribosomal RNA. Okay. We need tRNAs, and so these are RNA three-dimensional structures, right? They're kind of form this sort of cloverleaf shape. I don't actually know what these loops over here do. Involved in something, I guess. But there's two main points of note in a tRNA. One is where the amino acid is attached to, right? So they're going to be like reasonably large, sticking out the top. I'm sorry, tired. And the second really important, and these two are very much kind of tied together in a sense, functionally at least, part of the tRNA is this bit down here, which is the anticodon, right? And an anticodon is essentially the bit which allows the translation to occur, right, between the information in mRNA and whatever amino acid that information encodes. And so that anticodon is 3 RNA bases, and it is the exact opposite of the codon for which it is carrying an amino acid. So it's hard to kind of stress this. I'll draw it out too. Kind of hard to put that in simple English that kind of makes sense. But essentially the tRNA is carrying an amino acid, and that amino acid corresponds to the codon in the mRNA. The way in which those two get paired together is via the anticodon, right? So e.g. start codon equals methionine. Oops, hang on a second. AUG equals methionine, right? And so tRNA met, right, which is the tRNA that carries methionine, has, well, I'm just going to make sure I get this right, UAC anticodon. So essentially, I know this is a little small, but I don't want to switch back and forth too much. Oops, hit return. So here's your mRNA. That'll be your start codon. Right, this is also here, for example. Here's your AUG. And then you'll have, let's see if I can, let's make it really big. Right, that's a really crappy looking tRNA, but you get the idea. You will have the opposite at the bottom of that tRNA, so that you get hydrogen bonding base pairing, essentially the same as you do when you get double-stranded RNA, double-stranded DNA, DNA-RNA hybrids, stuff like that. You'll get an exact base pairing between, well, typically it's exact, but especially between these two bases, but typically between all three, right? And so that's how you go from this information here, right, to that information there, right? This tRNA, this RNA molecule with its anticodon, is the intermediate, right? That's essentially what translates those letters or words into amino acids. Questions? I'll take a picture. All these pictures will go up like the other ones. So some of you are probably thinking, well, okay, great. You know, how do we get the right amino acid on the right tRNA, right? Because that's pretty important. If you end up with the wrong one on the tRNA, then you're kind of screwed, right? The whole translation thing isn't going to work. And so that's where, and it's a really long word, it's amino acyl tRNA synthetase there. Essentially what this is the enzyme that puts the amino acid on the tRNA in the first place, or charges the tRNA. So a tRNA that's just RNA on its own is uncharged, right? It can't participate in translation because it doesn't carry an amino acid. And so this enzyme here, this amino acyl tRNA synthetase, does that. And it does so using the anticodon, right? So essentially, it has a very particular shape, right? It holds the amino acid kind of as part of its shape, right? And only the tRNA that has the correct anticodon will be allowed to go into that enzyme and have that amino acid attached to it, right? And so that's how we get that specificity. So in a way, these enzymes, these tRNA synthetases, are as important as the tRNAs themselves when it comes to the fidelity of that translation process, right? It's accuracy, right? So again, these enzymes use that anticodon to make sure to make sure the correct amino acid is stuck onto that tRNA. Okay, questions? Anybody got any questions? Kind of. So there's an elongation video. Oh, here we go. Here. Gotcha. Hidden in this link. Where are we at? Oh, God damn it. It's so annoying how. Go away. Is it that one? No, that's for my alignment. Here we go. So this one, which is from that, I'll put this link in the chat. I don't know why kids back chat and rudeness came up on my YouTube feed. It seems kind of weird. Right, so this is the mechanism of elongation in general. Go away. And then we have this, which is a really nice, actually I haven't seen this one before. Ooh, let's have a look at this one. Oh, well, I really like these videos from WeHi. See, simple, isn't it? We'll look at that in a little bit. And we'll do it like slow down a lot. So those two, and there's actually another one, the translation video or central dogma one. I think this might just be a new version of that. I'm not sure, I haven't seen it before. But those are good videos. The elongation video is kind of really specific to prokaryotes, but it's a lot of the information transfers across. The WeHi video is a more generic one, but it's kind of more accurate and not necessarily, but maybe a little less easy to follow. Right. But this one has the other one I don't think actually has annotation, which is kind of crazy. But this one does. Right. So those are both good videos to watch. There we are. Here we go. Okay, so essentially those are the components. I'm just going back here for a little bit. These two, so these are the small and large subunits, right? And it's not specified whether they're eukaryote or prokaryote because they're largely the same. So there's a lot going on here, right? Just in this one kind of image. Okay, so just kind of break it down into slightly more accessible concept, I guess. Small subunit is involved with the mRNA. So the small subunit essentially is involved in kind of moving along the mRNA. It's kind of like the motor, essentially, right? Or ratchet. Ratchet is probably a better word. And so it doesn't move smoothly. It goes in jerks of 3 bases at a time, which are those codons that we talked about. Does everybody understand what a codon is? Because otherwise it's going to be really confusing. If you don't, just let me know, like privately if you want, if you're worried about, you know, what other people might think and stuff. But anyway, the large subunit is all about controlling entry of charged tRNAs, formation of the peptide bond, right? And this out here is where the growing polypeptide chain is going to be pushed, right? Right. So entry of charged tRNAs, formation of the peptide bond, and exit of the uncharged tRNAs, the used up tRNAs, right? And so that's really kind of what these 3 sites are all about, right? And there's actually a region in here, what's it called? Aminopeptidyl transferase or aminoacyl transferase, I can't remember its name. That is the ribozyme that catalyzes that process, right? So you have the little motor along the at the bottom, which has the mRNA going through it, and then you have the kind of the enzyme part on the top, which does the whole making of the polypeptide chain, right? And it has a very specific direction. As mRNA has direction, proteins have direction. There's a very specific flow of stuff, right, essentially, right? And so in this, looking at this picture, it's going to go to your right, right? And so tRNAs are going to come in on the right into that access site or the A site. I call it access. The fancy word is amino acyl tRNA binding site. So access is a lot easier. Once it's in there, there's a relocation or translocation perhaps of one codon. A peptide bond is formed, so another tRNA comes in, and then a previously used tRNA exits, right? So there's always stuff coming in, bonds being forming, stuff leaving, right? And it's a constant flow through of tRNAs, leaving behind those amino acids that basically get kind of extruded out the top, like, you know, you're icing a cake or something like that. Okay. So again, a lot going on here. Once you get the general idea of it, it's fairly straightforward, but the details can be potentially overwhelming. And we'll have a watch of those videos in a little bit. I want to cover the basics first so that you have the context, right, with which to understand the videos, because otherwise, you know, you're not going to get the full benefit of it. Okay, so initiation, right? We need to start this process, right? And this is for eukaryote, you can tell, because it has 5' cap, even though that's not implicitly put there. We have cap binding proteins, so they have to be. We have poly A binding proteins in the tail, and essentially those coordinate to stabilize or recruit and then stabilize the ribosome here. So that's like the general conceptual view, right? Remember I talked about how the 5' UTR and the 3' UTR are important in translation, right? So you can interfere with this process, right? And so you can downregulate gene expression even if you have the mRNA there by affecting translation. Okay, I'm going to start with prokaryotes first. Ooh. I'm sorry, I'm really tired. Okay, so again, we've got a large subunit, we've got a small subunit. Again, the small subunit, so don't worry about the numbers. It's 60 and 40, I think, in prokaryotes rather than 50, 30 and 60, 40 in eukaryotes, 50, 30 in prokaryotes. The numbers aren't super important. It just tells you that one's bigger than the other one, right? And so essentially what we need to do first is to get that small subunit bound to the mRNA, right? So here's one big difference between prokaryotes and eukaryotes. Prokaryotes bind directly to the start codon, right? You can see it here. Well, they don't bind to the start codon. What they bind to is what's called the Shine-Dalgarno sequence, right? Which is that bit over here, just upstream of the start codon. And so that is, it's kind of like the equivalent to the Tata box, right? That we talked about in transcription, where RNA polymerase binds in the core promoter. This is kind of the equivalent for translation, right? And so two things. One, we have an initiation factor called IF3. Its job is to stop those two ribosome subunits sticking to each other, because we don't want that to happen yet. We've got to find the start codon first, right? So that initiation factor stops that from happening. Then this small subunit finds the Shine-Dalgarno sequence and sticks to it, right? So again, in prokaryotes, it's very directed, right? This is where we're going. We know what we want. And then we begin the whole assembly process, right? And so again, this is a sequence of events, and they happen in particular order, and there are particular things that facilitate that assembly. So we've got the small subunit bound, right, to the Shine-Dalgarno and to the start codon. Now we need to bring in our methionine tRNA, right, and some other bits and pieces, right? So that's what we're going to do next, right? We bring in our MET tRNA or tRNA MET, and it depends on how you want to write it, and we bring in a bunch of other bits and pieces, right? One of which is GTP, right? So remember again, GTP is essentially the energy that drives translation. This is one reason why, when energy levels are short, you start shutting down cellular metabolism, right? Because you don't use, you don't have the energy to make more proteins, or you might even start breaking down the proteins you have in order to generate that energy so you can make new proteins, right? So it's kind of like an interesting tie into like the bigger picture, right? So this takes a lot of energy to do. So another initiation factor comes in. This is IF2 with GTP, and basically the energy in the hydrolysis of GTP, right? So, and this is very much the same as it is for ATP. Let's see if I can do arrows. Goes to GDP plus energy plus what's called pyrophosphate, which is essentially just free phosphate, right? It's the kind of bit that's broken off that goes from being guanidine triphosphate to guanidine diphosphate, right? So GTP is hydrolyzed. That provides the energy to kick all that other stuff off. Remember, we had IF1 at least. Remember that one there. Its role was to prevent the binding of the large subunit. So GTP hydrolysis provides the energy to kick those things off. Now the large subunit comes in, and it fits right over the top of the methionine tRNA. Let's make sure I took that picture. We did. So I'm just going to draw this a little bit smaller. Let's stop the share so you can see what I'm doing off. Well, I'm not sure it pens anymore. That's a plus. So essentially you have your actually, let's do a little bit further down. I don't know what the next ones will be. Right, so those are our codons basically, right? Those are the words. So our Here's our SD sequence. Remember, Chi Nogado, not South Dakota. And that's our small subunit. Right. Once that GDP hydrolysis has occurred, right, and kicked all the other bits and pieces off, the initiation factors, then we get our, oh, hang on, let's do a proper job of our picture making. Ooh, I have blue. I actually picked up blue just so I could have more colors. Right. So before GTP hydrolysis, this is kind of what it looks like, right? The small subunit is bound to the mRNA. We've got our T met, our tRNA methionine is present. Yeah, hello, kitty cat. And now, once GDP hydrolysis occurs, we can bring in the large subunit, right, which is big, like so, right? And now we have our tRNA methionine is exactly in kitty cat, the P site. Oh, I just lost my pens. All right, so again, this is our A site. I like to call it access, just because it's easier to remember. This is our P site. That's our peptide bond formation site. And then this is our E site, which is our exit. Right? Stick that a little bit further back there. So when that large subunit comes on, and this is actually the same with, yeah, this is the same with prokaryotes. Let me just double check, eukaryotes rather. Yeah, that's right. I don't know why I thought it was different. When the large subunit assembles, this existing tRNA that's already there will slot very neatly into the P channel. Right, I'm going to leave this up here because I'm going to use that in just a second. Anybody got questions? Nope. Okay. So the big difference with prokaryotes and eukaryotes, right? Prokaryotes, the small subunit goes directly to the start codon. In eukaryotes, come on, we still have a whole bunch of initiation factors. Those are the IFs again. But here what we have is that the small subunit assembles at the 5' cap, right? So these initiation factors they recognize the 5' cap, right? And that's how they promote the assembly of the small subunit on the mRNA. So essentially these proteins recognize the cap. Then we have another bunch of proteins that bring in the small subunit. These are these EFs down here. And with them, they bring in a tRNA that's charged with methionine, right? So the big difference here is that tRNA is used like a barcode scanner, right? And essentially the small subunit will ratchet along the mRNA, or the mRNA gets pulled through it. Doesn't really matter which way around you think of it, until that anticodon of the tRNA finds the start codon, right? Does that make sense? Let's bring up the chat again. No, chat no want to work, no. Oh, for whatever reason, chat won't pop up. You'll have to shout it out if you have questions. Okay, so Cat, what are you doing? So Cat is learning genetics as well. I have to boot her out of here in a second. So anyway, what happens now is it kind of runs along, it processes along the mRNA until it finds the start codon. And as soon as it does, essentially the same thing happens as with prokaryotes. The large subunit comes in and binds to the small subunit, displacing or as a result of the release of all the other factors, right? And that tRNA, again, also goes into the P site. Okay, so that's the initiation. Big difference is prokaryotes find the start codon directly via the Shine-Delgado sequence. Eukaryotes find it by scanning along, like going, where is it? Where is it? Oh, there it is. Good stuff, right? And then they both have the assembly of the ribosome as a whole, right? So it's very much a stepwise process in both. Okay. Then what happens? Well, this is where it gets, let's see. That's not going to help. Oh, yeah. She's a very curious cat. Like all cats, really. So the fact that the tRNA is in the P site, the initial one, the methionine one, is really important, because that leaves the access site empty. Pardon me. And so now we get the next tRNA entering. Now that's supposed to be a G. Let's just write that a little more clearly. I don't actually know what amino acid is encoded for by GCC. You can look it up if you want. It's not super important to know that stuff. I've done this for decades and I don't know what. The only ones I know are the start codon. Actually, that's lysine. I don't know why I remember that, but you know. And then the stop codons are really important. So essentially, we now have another tRNA come in, and that tRNA is going to have the same principle of its anticodon must match directly the codon that it's going to be binding to, right? So if this is GCC, question for the class, what's the anticodon going to be for the next tRNA? Damn, that's fast. So it's going to be CGG, whatever that is. Oh, damn, I'm going to actually have to look it up now. Give me a second. Where is it? Come on, G, C, C. Oh, that's alanine. The problem with being a pedantic scientist, I guess. So that tRNA is going to be T-ALA, right? It's going to be the tRNA that is charged with alanine. So now there's going to be two steps. One, we need to get that amino acid off of its tRNA, right? It's bound, covalently bound. There's no like ionic or hydrogen bonds involved here. It's physically like bound via covalent bond to its tRNA. So the first thing that the peptidyl transferase, sorry, my cat just found a pencil, does is it breaks this bond here, right? So it breaks the tRNA AA bond, right? That's the first thing that ribozyme does. Now, obviously, the amino acid isn't then allowed to kind of, wander off, right? Because that wouldn't work. But the next job is, I don't know, hang on, let's do this properly. So 1 entry, I was getting out of step, sorry about that. 2, the tRNA in the P site has its amino acid removed from it. And then 3, we basically, I'm just going to draw a single line, right? That's how simple it is. Ta-da! Did you miss that? I'll do it again. Ready? Just so you're kind of, bated breath. There you go. That's it. And 3 is peptide bond. Peptide bond formation. Now, as a result of that, this, I'm not going to draw it all out because I just don't have the space on here, unfortunately. This tRNA is now uncharged, and we need to get that out so that we can get another one in, right? Which will be the tRNA lysine. There you go. Lysine tRNA that will correspond to this codon. And so now we have, I guess we could have another little step down here. That's going to be 4 GTP hydrolysis, right, is the next final step, and that moves the whole shebang down 1 codon, right, or along the mRNA by 1 codon. And so now you just have to kind of imagine this a little bit, because it would take too long to draw this out all over again, right? This uncharged tRNA is going to be in the exit site. This tRNA, which now has the 2 amino acids attached to it, is going to be in the P site. And now the A site is going to be open. It's going to be empty. And so now the next tRNA can come in, right? And essentially you just repeat this whole process again. It's very iterative, right? So you get the next tRNA come in. You'll then have this dipeptide, right? This met-ala will be removed from this tRNA, which is actually going to be here. It will get added to the new amino acid that's just come in, the 3rd one. GTP gets hydrolyzed, and we move down 1 more word or codon. That's basically translation. Don't know what she's doing. Got any questions? Ta-da. So now you can also see how directionality of DNA, right, equals directionality of mRNA. Directionality of mRNA, as you can see here, now equals the directionality of an amino acid, of a protein, right, polypeptide sequence, right? So it flows through from one to the other to the other. All right. Okay, so let's take a picture of that before I forget and erase it. So let's see, let's have a look at that WeHi video, which is pretty neat. Oh yeah, so this is how I found out that GCC was alanine. You can see it just down here. All right, let's see how well. And can I? Oh yeah, I can slow down the speed. Let's have a look first. I'll slow it down a little bit, don't worry. Well, that's a little bit too short. Let's go find the other one. That thing at the end looked like something out of the Final Fantasy film, which is kind of neat. Don't know if anyone's seen that. Here we go. This is one that I've used before. All right, so let's slow this down. Touch. So what you're seeing here, this is the nucleus or nuclear membrane. These are nuclear pores, right? This is how stuff gets into the nucleus, such as proteins, right? Because that's what, you know, DNA polymerase, for example, has to get in there somehow. And it's also how stuff gets out of the nucleus, such as mRNAs. And there's not really a huge open space in there. It's jam-packed with stuff. It just makes things easier to show. All right, so this must be a prokaryote by the looks of it, because there's no scanning involved there. Let's get a little bit further along. So here you can see they're all kind of like color-coded the same. Here you can see that ratchet process, right? Where these go in jumps of 3. Right? And that's what those codons are all about. And in the background, you can see the tRNAs kind of flown around with their amino acids stuck to them. Okay, so let's have a look at, so essentially that light blue part down at the bottom, that's your small subunit. This large kind of purplish part at the top is your large subunit. Oh, look, they've got a spliceosome video. I should watch that. Okay, and even though they don't have, so this is where it falls down a little bit because it doesn't have any kind of narrative, right? So it's kind of why I want to take you through it. But essentially, we're going from right to left, right? So remember I was saying that there's this flow through of stuff through the ribosome. We've got charged tRNAs coming in on the right, and you can just see one peeking into the frame on the right there. And we have uncharged tRNAs or used tRNAs exiting to the left, and the whole thing moves to the right, or the mRNA moves to the left, whichever way you want to think about it. And so here we've got that same deal in terms of sites that you can see, yeah, where are we at? Here. right? We have the access site on the right, we have the P in the middle, or the peptide bond forming site, and we have the exit site on the left. So you can see here we have a charged tRNA that's come in. Here is our tRNA with all of those amino acids, right? This is that red thing is that amino acid chain. All of those, that polypeptide chain is attached to that one tRNA, right? And so, and then on the left, we have a used up tRNA. You see there's no red bobbing on the end. That's exiting from the E site, right? So again, the access site on the right, where the tRNA comes in, the P site, which has the current polypeptide chain attached to the tRNA, and the E site, where that used up tRNA from the previous round gets ejected. I don't know why I can't bring up chat when I'm sharing. It's really, really annoying. That doesn't help. Okay. So I'm going to slow this right down. Okay, so you see that charged tRNA coming in, right? This chain gets stuck to it, and then it gets stuck to this one, and it gets stuck to this one, then it gets stuck to this one, and it gets stuck to this one, and that used tRNA is leaving, right? So it comes in, gets swapped over, comes in, gets swapped over, comes in, gets swapped over, so on and so forth. Right, this is just kind of showing more of the same. This is essentially the pocket into which those tRNAs enter, right? So let's just show you that again. This is why I have to slow down like crazy, because it's, what's that, 13 seconds? It's not a lot of time to look at that, right? So you can see just there, that red bob, you know, I like think of it as like the bobble on top of a Santa hat, right? This whole chain is a series of amino acids, right? It's the polypeptide chain. Here's the next amino acid that's going to get added onto that. So one would think, oh, okay, well, we're going to take that chain and we're going to add the amino acid onto it. Actually, it's not how it works. What you do is you bring in the amino acid and then you stick all of the existing chain onto the front of that new amino acid, right? It's not like DNA replication or transcription where you have a chain and you add stuff onto the back of it, right? In translation, it's kind of weird to think about it, but you're actually taking the existing chain and you stick it on to the front of the new thing that's coming in. That moves over, and then you move the whole thing onto the next one, and so on and so forth, right? So it's not so much that this amino acid gets stuck onto the end of that chain, it's that the chain gets stuck onto the amino acid, right? In that transferase process from this tRNA in the P site to this new tRNA in the A site. And you can see that as it comes in, like so. Let me put that, I'm gonna have to stop sharing, unfortunately, because I can't access the chat. I don't know why it does that, it's very variable. So there's the other WeHi video. It wouldn't hurt to watch all three, to be honest, so because they all have their own different strengths and weaknesses in terms of the information they're able to get across. And then I'm going to take a break now because I'm pretty pooched, and I'm sure you are. When we get back, we'll finish up by looking at these things called polysomes, some funkiness, essentially. Oops, hang on, I'm not sharing. get back here. Right, we're going to look at polysomes and some funkiness there in prokaryotes, both in the structure of how their genes are organized and also the fact that transcription and translation is linked in prokaryotes in a way that it can't be in eukaryotes. And then at the end, we'll talk a little bit about the genetic code and some degeneracy and wobble, right? So I'm going to pause the recording for now. Let's get this out here. Let's meet back in, I don't know, 12:30, right? So we can get through the rest of it. All right, ladies and gents. Let's get going. Okay, so essentially what we've done there is look at the elongation part. Oh, I also forgot one last thing before we get on to any of the other funky stuff. And that is, how does this whole process stop? Right, because again, you know, at some point, oh, now I can see the chat. You know, you're going to run out of mRNA, right? And that 3' UTR is called a UTR. An untranslated region for a reason, right? Because it's not translated. And so actually you can see it here quite nicely. There are 3 codons which do not code for an amino acid, right? So they are not recognized by a tRNA. And they are here in red. So they are UAA, UAG, and UGA. And you can also see that back here as well. Right. Those are called stop codons, right? And so as soon as the A site moves over a stop codon, there's nothing to come in there, right? Nothing, well, there's no tRNA that will enter that site. Instead, what we have is what's called a release factor, right? And this is the same kind of principle as for stopping RNA polymerase, and in transcription termination. But essentially, this release factor comes in to the A site when that A site is over a stop codon. And via GTP hydrolysis causes a bunch of different things to happen. One, that polypeptide, which you can see here, polypeptide chain, actually it's attached to this one now. It needs to be released from the last tRNA that it's stuck to, right? And so we need GTP hydrolysis to provide the energy to do that. And we also need to get the ribosome, the small and large subunit, off of that mRNA, right? So that another one potentially can come along and translate that mRNA, or so that that ribosome itself can either go back to the beginning or start on another mRNA. It doesn't really matter, right? They're constantly being reused, right, over and over again. And so the release factor prompts the breaking of that bond and also the dissociation of the ribosome from the mRNA. So you end up going back to the constituent parts that you were at the beginning, right? That's the role of stop codons, right? And now hopefully you can see how much of an impact a stop codon can have on protein structure and function. If a mutation, whatever type of mutation that may be, frameshift or a nonsense mutation, causes a stop codon to be created in that frame. And so when we talk about frame, we're talking about the series of 3-letter words, the codons, basically. Because as soon as the stop codon is there, that's the end of the protein. Right, and actually one of the the genes I've worked on over the years, which is involved in metabolism. One of the mutant alleles that I worked on for many years has 7 amino acids. It has a nonsense mutation near the end of the last exon, which truncates it by 7 amino acids. That's it, 7. And it completely does not work, that protein, right? It's almost exactly the same size and shape, but those 7 amino acids are required for its enzymatic function. So without them, you have a loss of function mutation and a whole bunch of other stuff. Okay. Anybody has any questions, just pipe up. We're doing pretty well with regards to time. Okay. So one thing is that whenever we do these, watch these videos or we look at these slides, we always kind of present it like there's one mRNA and one ribosome, right? So the ribosome binds, it goes along, makes a protein, maybe the process starts again, right? It's a very kind of limited process, essentially. And that's not how it works at all. What you actually have are what are called polysomes, right? So as soon as the AUG is, so this is like the initiation, right? Essentially, right? You have the small and large subunit assemble, you start producing your polypeptide, and as soon as that ribosome is moved off of, right, it's a large protein complex, right? As soon as it's moved a ways downstream, right, and made a reasonable size polypeptide chain, like 25 amino acids, essentially it clears the space over the start codon. So another ribosome can assemble, and then it begins its process, and it kind of goes along, right? So it's basically like this series of, kind of trains like running along the same track. And you can have multiple ribosomes translating the same mRNA. So if you think about it in terms of, hey, we need to get a lot of something, whatever that may be, I don't know, antibodies made very quickly, right? Rather than doing one protein at a time from one mRNA, now you can have one mRNA and you can have multiple proteins being produced at once. So it's a very effective way to scale up protein production very quickly as necessary. And that's essentially what is called a polysome, because it's a polyribosome, basically, right? Okay, so what I want to do now is we'll get to the genetic code in a little bit. But what I want to do is talk about some of the differences between prokaryotes and eukaryotes in terms of translation. And this really comes back to what is a prokaryote and what is a eukaryote, right? Prokaryote, single-celled organism, no nucleus. Eukaryote could be single-celled, could be multiple-celled, doesn't, change the fact it's a eukaryote. What do we have? We have a nucleus. So in eukaryotes, transcription and translation cannot go together, right? You have to transcribe the DNA, process the mRNA, export it into the cytoplasm before it can be translated, right? So you can unlink the two. I mean, they are largely, they go together, right? Because that's just how things work. But they are not physically linked in time and space because of that nuclear membrane, which kind of acts as a dividing line between transcription and translation. Prokaryotes don't have that same barrier, right? So that's one big difference. And that is that transcription and translation are linked in time and space in prokaryotes. The other one is that prokaryotes have a different way of organizing their genetic information, right? Hang on, let's just want to get this rubbed off in case I need it. So typically eukaryotes, they have one gene and all of its gubbins, you know, regulatory stuff is one unit. That's called a cistron. Where's that definition? Here we go. Right. So euks are essentially monocistronic. Right. So what that means is, you know, one piece of DNA. Oh, yeah, eukaryotes. Sorry, it gets really tedious typing out eukaryotes and prokaryotes all the time. So I shorten it to euks and prokes. Hopefully it's self-evident. Right. Prokes or procs are typically polycistronic. And what that really means is that rather than having, you know, one gene for this enzyme in a metabolic pathway being on this chromosome, and we have one gene for the next enzyme being on a different chromosome, they're all over the place, right? And they're coordinated by transcription factors. In prokaryotes, typically you have functional units. So if you need to metabolize, I don't know, say a food source of some type, whatever that might be, and we'll look at some examples next week. All of the genes required to produce the proteins that can utilize that food source are together. So for example, you need, like for glucose, right? You need to get glucose in the cell, right? It's a large polar molecule. It won't get in there on its own. So you have a glucose transporter. Then you might have an enzyme which adds phosphate onto that to turn it into what is it? Fructose, right? And then you'll have another enzyme which is needed to cut that into 3 carbon sugars, right? And then another one, and so on and so forth, right? And so you need all of those to be able to metabolize that one thing. So there's no point just turning on one gene, because even if you bring, like if you express the transporter, it doesn't matter if you don't have the enzymes to metabolize that into something useful. And so prokaryotes instead lump all of those different reading frames or coding sequences, or even genes, I guess, you know, kind of a more of a nebulous term than we think, all of those together so that you just turn on one thing. Glucose is present. Yay! Everybody loves glucose. Produce all of the proteins that are necessary to metabolize glucose, right? So basically, they're linked in groups called operons. Oops, if I can spell single. Functional purpose. Okay, glucose metabolism. Metabolism. Right. So these produce what are called polycistronic mRNAs. So this is quite a nice image down here, right? Instead of in eukaryotes, you know, you produce one mRNA per gene, right? And then you translate that mRNA. In prokaryotes, Instead, you produce one large, right? Remember, we've got all these genes kind of like stuffed together into one unit. We're going to make one mRNA, like for the whole lot, all of those genes. But that polycistronic mRNA, as you can see here, has, in this case, 3 reading frames or 3 coding sequences. Right, we have our first one here, so a ribosome will come in, it will assemble, translate that, produce a protein, might keep moving or it might dissociate, come back somewhere else. But it will then, it or another one will be able to produce the second protein from that polycistronic mRNA, right? So all of the information necessary to make all of the proteins needed for that one process are on one mRNA transcript, right? So in a sense, it's a way of coordinating gene expression. Rather than having to put together all these different genes and make sure that they're turned on at the right time and at the same time to do one thing, instead in prokaryotes, they're all lumped together into these functional groups, right? That's definitely not the case in eukaryotes, right? In eukaryotes, they are scattered, all over the place in different chromosomes in different places. And instead, they're coordinated by the use of transcription factors, like proteins which control transcription. So it's just a different way of doing things, right, essentially. Does that make sense? Any questions on that? Oh. Okay, so the next oddity between, or difference, we shouldn't call it oddity, difference between prokaryotes and eukaryotes is that in prokaryotes, as soon as that mRNA becomes available, right? So it essentially the start codon or the first start codon emerges from the RNA polymerase. A ribosome will assemble on it and start translating it. And so transcription and translation are linked in prokaryotes, or coupled. There you go. That's probably a better way, right? And so essentially, you have your DNA, and this is what it kind of looks like in real life, right? When it's kind of extracted from a bacterium. As soon as that DNA, that gene or that operon starts being transcribed, as soon as that transcript is long enough, an mRNA will, ribosome, sorry, will attach to it and start translating it. And so you can have, from one single operon, you can have multiple transcripts. Each of those transcripts is going to have multiple ribosomes assembled on it, translating the protein from that particular operon. So again, it's a good way of scaling up the issue. In bacteria particularly, because they're much less able to move from environment to environment the same way we do. They have to be able to change themselves, essentially, in responses to changes in their environment. I don't mean like changing their structure, but changing their response to that environment. If we're cold, we go somewhere warm, right? If bacteria get hungry, they can't go to the pantry. They might be in the pantry. They have to change their metabolism based on that new environment. And so this coupling, right, both polycistronic mRNAs, the whole polysome deal, and coupling transcription and translation allow that to happen very fast and in a very coordinated way, which is particularly neat. This also has a lot of consequences when it comes to control of gene expression, because depending on how fast or not that translation occurs, which can depend on presence or absence of particular metabolites or amino acids, can control whether or not that translation occurs at all, right? Or even the gene expression in terms of transcription as well, right? So this coupling is actually really important in terms of control of gene expression as well. I mean, it's fairly straightforward of a concept, but it's a very important one when we get into gene reg later on. Okay, so really all we've got left to do now, we actually will have some time at the end if you wish to use it for any kind of recap before the exam on Thursday. But all that's really left is the genetic code and degeneracy. I'm not talking like moral or ethical degeneracy. We're talking about degeneracy in the genetic code, right? So essentially, that's the genetic code, right? What this is a kind of interpretive table, right? And basically what it tells you is what codon equals what amino acid. So with a few rare exceptions, and that's really down to wobble, which is kind of a fairly nebulous concept, one codon equals one amino acid. Right, this is an important distinction to get because it doesn't go the same the other way. This is why you can't go from protein back to mRNA or DNA, right? One going from RNA to protein, one codon equals one amino acid, right? So UUU will always be phenylalanine, right? It'll never be serine, it won't be arginine or leucine or whatever. UUU will always be phenylalanine. Same as AUG is always methionine, right? And that's pretty much the same for every organism with a few rare exceptions, right? So in terms of information, mRNA to protein, one codon equals one amino acid. Doesn't work the same way backwards though. So one amino acid can be encoded by more than one codon. So essentially it's like different words that have the same meaning in language. Is that homonyms? Yeah, I think so. Not very good at grammar and stuff. Yeah, synonyms are ones that sound the same but mean different. Homonyms sound different but mean the same, I think, from what I can remember. Right, so essentially these are homonyms. So where's my favorite? Leucine. Here we go. So leucine, for example, which you're looking at here, there are 6 different codons that encode leucine. So wherever you see one of these 6, you will find a leucine amino acid in that polypeptide chain, right? Now you've just got to change one of those letters, right? Could either be the first letter or the last letter, and you will get a different amino acid, right? Because again, one codon equals one amino acid, right? Because that's based on the tRNA. So that codon will only be recognized by and large by one tRNA, right? It's not going to be recognized by a tRNA. So if we have UUC, for example, it's not going to be recognized by the codon which encodes serine, UCU, right? Or even UUU, right? Even if there's one base difference, right? But each amino acid can be encoded by multiple. So that means that the information can't flow the other way, right? That's the whole point of the central dogma. Because if you have a serine in an amino acid, well, you could have 4 different combinations of 3 letters present, right? They're all going to be U and C, but it could be U, C, A, or G in the 3rd position. You've got no way of knowing, right? So that's why you can't go backwards from protein to mRNA. You can kind of do it in terms of cloning, because then you use what are called degenerate primers, right? So they have any number of bases at that particular place in the primer, right? So you can use a protein sequence to clone a gene, right? but you have to take that degeneracy into account when you design those short bits of DNA to amplify that gene. Which also means you get a whole bunch of other random nonsense as well, which is really annoying. Okay, so that's what we mean by degeneracy, right? And that's also really important when it comes to mutation too, because you can see for leucine, for example, if you have a mutation in the last letter of that codon and the last of the three, it's not going to have any effect, right? You could go from CUU, you could have a mutation which goes to CUG. Doesn't matter, you still have leucine, right? So that's why many, not all, but many of the mutations that occur in codon sequences are silent, right? They don't have any effect on the protein sequence. And so in some way you can think about this as kind of like built-in robustness, right? Because many proteins are utterly essential, and so if they become non-functional via mutation, then that could lead to the death of the organism. So in some sense, this is a handy, might be just a byproduct, but a happy consequence of that degeneracy is that it makes codon sequences much more robust, right? Now, obviously, if you have, so here's one which is not going to help you out, right? Tyrosine, I think there's only two. Yeah, I think there's only two for tyrosine. If you have a tyrosine codon at UAU, and that last letter changes to a G, right? Now you have what's called a nonsense mutation, or an introduction of a stop codon. So now that tyrosine codon becomes a stop codon, and now your protein translation ends at that new mutation, right? So it's not perfect, that's for sure, but it gives a little bit more kind of robustness into the production of those proteins. And actually, in terms of like real big picture stuff, like evolutionary biology, when you look to figure out how old an organism, a species is, or when its last common ancestor existed, you typically use very conserved protein sequences to do so, or ribosomal RNA sequences is the common one, right? Because those do not change very much at all. Their mutation rate is very low, not because mutations occur less frequently, but because those mutations never persist in the population, because they are typically lethal, right? So if you want to figure out how old, like the last common ancestor was Sasquatch, right? If you're Canadian, right? Or Yeti if you're Alaskan, maybe. Then you would use one of those very conserved, very essential genes, and you'd look at the rate of change and mutation in them, and that will give you an idea in terms of, are we talking hundreds of thousands or millions of years ago kind of deal, right? So that's essentially one way in which they figure out when, humans and Neanderthals were in contact in Europe, for example. They're able to sequence Neanderthal DNA and look essentially how that has changed in humans over that time span. And also the fossil record and stuff like that, which is kind of cool. So anyway, that was a little bit of a tangent. So essentially degeneracy is that the fact that more than one codon can encode for the same amino acid. Wobble is slightly different. What that is talking about is that actually it's not 100% true that only one tRNA binds to one codon. There is a little bit of wriggle room, particularly in the last base. So the first two are very important, Sometimes in the last one, any will do. And that kind of ties into this degeneracy, right? So if you have a tRNA for leucine, for example, right? A tRNA with a leucine tRNA with an anticodon of, what would that be? GAU, right? That third one down here can still bind to a codon, which is CUU, right? So it'd be GAA would be the anticodon. And so that kind of just makes things a little bit more efficient in the sense that if you're a little bit short of charged tRNAs, so leucine tRNAs of a particular sequence, you can use the other ones basically, right? So that's kind of one way of thinking about that. Obviously, you don't want that to happen all the time because otherwise you could end up with all kinds of crazy shit happening. Like you can have all wrong amino acids introduced into that polypeptide sequence. But typically when you have, oh, serin's the other one. There you go. There's some serines down there. When you have amino acids that can be encoded by 4 codons, and they all share the first 2 bases of that codon, then the stringency is much less when it comes to whether or not that third base matters, right? And so that's basically what we mean by wobble, right? And that's pretty much it. So that's it in terms of material for what we will be covering in the exam on Thursday. We do, if you wish to use it, have some time to go over anything from the material that we've covered on Thursday or today. Oh, quick question actually. Has anybody been able to, oh yeah, and also transposons and stuff like that. I just don't want to forget those. Oh, there you go, all this stuff. Has anybody been able to access the feedback from exam 2? Because I know that some of you had issues and I'm not sure if it was actually resolved or not. Has anybody had a look at their exam 2? Okay, Peyton, did that change from exam 1, or you always able to access the feedback? Okay. Sometimes, I don't know if it's the case in this instance or not, but I know that occasionally students have issues with accessing Blackboard on their phones. So I don't know what you're using to access Blackboard regularly, but sometimes working off of a laptop, for some reason, I don't know why. Yeah, again, sounds like that's not the issue here, but sometimes that can cause problems too. Oh, you definitely have to wait until the due date passes, Peyton, without a doubt. And that's really deliberate so that you can't complete the exam early and then pass on the answers to someone else. So that's kind of baked into the setup. I have 2 exam 1s on there, no exam 2. Pretty sure you did exam 2. Is it mislabeled? Oops, sorry. Hang on, let me get off of here. Oh yeah, sorry. That's let's see if I can change that. Oh, come on, Blackboard, you pile of turd. So I'm just getting back into Here we go. Yeah, that's a funny mistake to make. Well, hopefully everybody found the right exam to take. No, could go back. What a silly mistake to make. All right, let's see if that's changed it in. That should hopefully change it in the grade center too. Sometimes I feel like I'm a purveyor of arcane information whenever I try and interact with Blackboard. Exam 1. Yeah, that's changed it. Thank you, Thank you for that, Peyton. I never realized. Sometimes I'm just so happy that I finished doing the exam, like, yeah, finally I can, put that off my list and do something else. That I should just check that a little bit more thoroughly first. Well, at least now that, if there's another second, like exam 1, you'll know what the issue is. Yeah, I'm sorry about that. Sometimes I just make silly mistakes because I'm in a rush to get off and do something else. I'll double check for this next one though. Yeah, so Angela, have a look at the now exam 2 and see if you can access that. Okay. All right. So does anybody have questions, clarifications, stuff that they want to go over for the last, I don't know, like 20 minutes or so? Again, we don't have to, but it's not going to hurt you to do so. You can't open the past test. Okay. Andrew, if you could do me a favor, it's partly to help you and partly so that this doesn't happen again in the future. If you could just send me a quick email with what you have done and the fact that it doesn't work, then I can forward that on to Cece, who's our Blackboard expert, because she, for whatever reason, it's just not working with you. And I haven't done anything different from what I've done in the past where it has worked. And so it'd be good to figure out what additional quirk of Blackboard is, causing issues this time around. Because it is a funky ass program, that's for sure. Ooh, probably shouldn't say that on the recording. Exam 3 is over 12, which is eukaryotic genome structure, 13, which is transposons, 15, which is gene expression and transcription, and 16, which is RNA processing and translation. So it's PowerPoints 12, 13, 15, and 16. There's no 14. It's like that missing floor. Oh, actually, I should have done it for 13. Never mind. It's like that missing floor in the elevator that you just can't get to. Okay, questions, peeps? Oh, you can study for a final, Angela. You just haven't covered that material yet. Oh, okay. Yeah, I mean, hopefully we'll figure out. If I have to, I'll just print out copy of them. I'm a little bit leery of putting electronic copies of exams out there, but also we can do what we've done before, which is just go over each exam in turn. I don't mind doing that. If we want to do that, today would probably be better than tomorrow, because tomorrow I'm due to be out and about. Yeah, I know. I understand that. I appreciate that. But yeah, send me that email. And Cece is actually really good. She's really into what she does, and I'm sure she'll be very keen on trying to figure out what's going on too. So as soon as you can, send me that, and I'll get back to her about it. Yeah, well, we can talk about that privately and later. If not today, then like by email or something like that. I'll figure out a way of making sure that you're not missing that information. That's for sure. Okay, anybody have questions? Questions? All right. Well, if you do have questions, just bring them to lab, right? You know, we have time there as well. So why don't we take an early break today instead, and we'll start the lab at 1:45. Yeah, let's get this. Okay, and then we'll go through what we're going to be covering in lab in about half hour or so. Give you time to have some lunch. Cool. All right. Thank you very much, everybody. See you in a little bit. Enjoy your lunch.